So hello everybody. Welcome to our third Ask Me Anything speaker series with our student author. Today we have Mr. Ricky Pimentel from the University of North Carolina. So we'll just take it away, Ricky. We'll have questions at the end. And then if there's any questions that people don't want recorded, we will end the recording to have more discussion off recording. There you go. All right, take it away, Ricky.
Can you give me a screen share ability?
All right, try it now.
Sweet. All right, can people see that? I see a thumbs up. Awesome. Okay. All right, sweet. Thank you for inviting me to talk about my research and just give me the floor to just talk about what I've been up to lately. This should be pretty fun. I hope it is. And yeah, I'm excited for the discussion afterwards. So thanks everyone for being here. Okay, so really briefly, just an agenda. I'll talk about myself a little bit so everyone maybe knows a little bit more about my background. Danny and Evan asked for 5 highlights about a recent study that I did. And so I'm just gonna like breeze through those and just stay pretty high level on those 5 highlights that you see listed there. And then they also asked me to talk a little bit about helping to get your experiment through to a publication. And so I'll just share some tips that help me to stay on track most of the time. That's a pretty hard way to, publishing is not easy. We'll just start there. Okay, so about me, I grew up in Michigan. I was born and raised in Flint, Michigan, and I was always like really involved in sports growing up. So baseball, soccer, football, hockey, lacrosse, pretty much any sport I could get involved with, I tried doing at some point, lots of dabbling. And then also as a kid growing up in the 90s and early 2000s, I was a huge Red Wings fan because they were really, really good. And they made the playoffs every single year I was alive until I hit like 25 or 26. And then it's been a rough patch since then, but hopefully soon they'll be good again. That's fair. That's fair, Kristen. What a good rivalry. What a good rivalry with the Avs. So, I mean, speaking of the Avs, when I was 18 and graduated high school, I moved to Colorado, where there's a lot of Avalanche fans, but I held my own. And I went to Colorado State, and I studied health and exercise science there with a concentration in sports medicine. And I got really involved in biking. And so that's been a huge part of my life for the last decade or so. Lots of bikes. But I wrapped up at CSU and I didn't quite know what I wanted to do. I knew I wanted to move on to grad school, but I think I needed a break. And so I ended up graduating in December. I took 4.5 years. And one of my buddies from undergrad got a job in Lake Tahoe. And he's like, hey, come out and like live with me for a little bit. And so I went and was a ski bum in Lake Tahoe, and I worked for Heavenly for 5 months, and it was super fun. And I recommend people to just go out on a limb and try something different, and maybe don't follow like the same like path as everyone else. Just go and be yourself and do your thing. And that was just a really good experience to like live somewhere new and try out some different things and see a new area. But then I got into grad school back at CSU, so I returned to Colorado State in Fort Collins and stayed in the same department, did my master's in health and exercise science, and that's where I started to learn some biomechanics chops. So I finished that up in 2016. Then I moved to Denver and worked for the Children's Hospital of Colorado, and I was a researcher in their clinical Gait lab that they have there. And that was a really, really good experience. Got lots of experience with motion capture, force plates, working with children with neuromuscular disabilities. Highly recommend if anyone is interested in the clinical biomechanics realm, these positions in children's Gait labs can teach you a lot and give you like a really broad experience in how biomechanics can help people out. But after 3 years of that, I was ready to move on to the next step, and there was an opening at UNC and NC State, and that's where I've been at for the last 2.5 years. And I've been studying metabolic cost and walking biomechanics, and we'll get into that more now. So first, I'm going to talk a little bit about biofeedback, and this is just a research tool that can be really helpful to investigate gait modifications. And here's a couple examples of biofeedback that we use in our lab. And last year I gave a talk that kind of like summarized more about biofeedback biomechanics and how it's useful. And there's mainly like 3 takeaways for that. And the first one is normalization. And so it kind of like gives everyone the same baseline. And so it'll like normalize the group to whatever their baseline is at. And then you can start to look at their adaptations from the biofeedback. And so I'm going to walk different from everyone else in this room. And so the normalization kind of gives you the same starting point. Whereas then once you start to implement the adaptation or the biofeedback parameter, then you're actually measuring whatever change you're looking to make in their walking pattern or whatever the outcome is. And then lastly, depending on how you build it, you can turn it into a game. And that can be a little bit more fun for the participants, especially if you have longer trials, which for me, I had 5-minute walking trials times 12. And so making it a little bit of a game definitely helped out our participants to kind of stay on task for the full research visit. But this is a really big and growing area for gait biomechanics. And so there's been a huge burst of studies in the last decade or so in gait biofeedback biomechanics in a lot of different populations, looking at a lot of different types of biofeedback, mostly visual, which is what we do in our lab, but auditory and sensory and lots of combinations of those. And then also you can change up the parameters. So you can have joint angles, you can have forces or joint moments, cadence, temporal spatial measures, EMG, and definitely more than what's even listed here. And so I would just like challenge the group to like think about what other forms of biofeedback exist and how can you incorporate that into your research. One that I always think about is like the 10,000 steps per day thing that we're supposed to strive for as an active human being. Like that's kind of a form of biofeedback. It's not quite on the same time scale as what we're measuring in the lab. But I would consider that a form of biofeedback, as well as a lot of different sports, like hitting a home run versus striking out would be a different form of biofeedback. Like they maybe performed almost the same motion, just maybe their timing was off, or they had a slightly different angle of the bat. And that's the difference between winning the game or walking back to the dugout. Same with swishes and air balls, all that kind of stuff. Like it's really fun to think about how different forms of biofeedback could be used for scenarios like this. And even shoe wear patterns, that's even like the longest term one that I was thinking about while I was making this presentation. Like that's days, weeks, months of similar patterns showing up on the bottom of your shoes. So that's all I have for biofeedback for right now. But I'm here to talk about this paper that was published last year. And I really have to thank these 4 folks here on the page, which are Noah Piper, Sydney Bowden-Tistel, Gabby Diaz, and Billy Clark, because I showed up in fall of 2019 and they just gave me this data set and said, all right, do OpenSim and do biomechanics on this and make it work. And so huge shout out to these 3 for collecting all the data. And then to Billy Clark as well for helping me wrap my head around how to use OpenSim, and lots of time banging our head against the wall trying to figure out how to make this work and like what is actually going on. But basically in this study, we are using propulsive force as our biofeedback parameter. And that is the horizontal portion of our ground reaction force during push-off phase of walking. And that propels us forward. And it's also paired with the braking forces of your leading limb while you're walking. So for our experimental design, we had 12 different young adults, pretty typical demographics, walking on this force measuring treadmill at their preferred walking speed. These were all at the same speed. And they were targeting these 5 different propulsive force intensities with visual biofeedback. And the short answer is any change in your propulsive force over time is going to result in an increase in net metabolic power while you're maintaining the same fixed walking speed. And this aligns with the theory that human movement is optimized to minimize metabolic cost or decrease muscle activation. So you can see as like whatever their preferred or normal value was the lowest cost for walking. And as they changed their biofeedback, changed their propulsive force, the cost of walking increased. Okay, so now we're gonna dive into some of the OpenSim stuff. And so if you haven't used it before, OpenSim is a pretty useful resource or tool to visualize, analyze, and process your biomechanics data. And basically, this is where your data can live in a simulation if everything goes as planned. So basically, at the end, we want to get this video or simulation of a person walking, and hopefully, depending on how far you go with your simulation, you can get muscle activations. And from that, I estimated the individual muscle metabolic costs. And so we have the empirical data that I showed earlier up here in the top left corner. But what we can do with these bioenergetic models in OpenSim is simulate that on an individual muscle basis and add that all up and see how that lines up with our empirical data, both here in our watts per kilogram, but then also we can correlate those, and we found that it explained about 33% of the variance between each of these models and the empirical measured metabolic cost. And the models were like almost exactly the same between the two. So we ended up using the Umberger model for the rest of this study. So just know that. But what the big thing that doing these simulation adds is that you now have a measure of timing. Like at every single frame that you're measuring biomechanics data, you can get that instant's worth of metabolic cost estimates across the gait cycle or across any time period that you want. And so you can't do this with your empirical measurement of metabolic cost. This is an average over the final 2 minutes of each of those trials. And so this is where OpenSim and the bioenergetic modeling really comes in handy. But there's a pretty big curve for learning and using OpenSim. I spent the first 6 months of my PhD just like learning about it and failing a lot before even like getting anywhere. And there's one point like early 2020 before the pandemic started, I was like having to go meet with my advisor, and I'm like, yeah, I still don't have anything. I don't know when I'll have it, but I'm working really hard on it. And it's difficult, but I would say it's worth it. But even once you do get it, it's still difficult because I still fail regularly. Even this last week, I was trying to run a bunch of simulations, and I was like, oh, I have all this data processed, like it's all good to go. We're good to go for this abstract. And we started analyzing the data more, and like these joint moments look way off. Like what's going on here? And we're like, okay, guess we can't use that for the abstract. We gotta go back and figure out what's wrong there. So it's a process, and there's a lot to learn and a lot to do. And yeah, just make sure you work with collaborators and always look at your data so you know, like, okay, this is where we're at, and we have some improvements to do, or you know, it helps to have those empirical measurements to line back up to your data. Be like, oh, this lines up with what we actually measured with oxygen consumption or another measurement, so you can kind of ground your results, which is very useful. Okay, but then in terms of this paper, I haven't really talked about why we want to be doing this. Like, why are we measuring propulsive force changes? Like, why are we doing biofeedback? And it's kind of all grounded in this theory of joint work redistribution as we age. And so my advisor and his advisor during his PhD were doing some biomechanics between young and older adult walking. And they found that the older adults pushed off the ground with less propulsive force and less vertical force during push-off phase. And this lined up with another theory that Paul DeVita and his group really grounded way back in 2000, where there's a shift in joint work from the ankle during push-off phase to the hip. And so older adults are walking with less ankle joint moments and less ankle joint power than young adults, and they're shifting that and doing more work on the opposite hip while they're walking. And this explained the higher levels of metabolic cost for older adults. This explained the higher levels of metabolic cost for older adults. And so we wanted to kind of dive into that more and see if that made sense from a modeling perspective. And the way we did that was we used biofeedback to steer young adults to walk similar to older adults. So we asked young adults to walk with a smaller propulsive force using biofeedback, and we found that they kind of do the same thing as older adults. So this can be used as kind of a model or a proxy for older adult walking, because when they walk with smaller propulsive forces, they shift joint moments to the hip, and they end up having a higher metabolic cost. And so we used OpenSim to break down these muscle costs, metabolic costs into different phases of the Gait cycle, which you can see here. But then we broke that up into all the different joints. So we looked at hip, knee, and ankle. And this is a lot of data, so I'm just going to highlight these two areas where the black line is the normal values and the dark blue is the smallest propulsive force value. And you can see that there's a lower amount of propulsive force or push-off force here from the ankle muscles, and they're shifting that to a higher metabolic cost at the hip at the opposite side. And so lower cost here at the ankle during trailing limb double support, which is push-off, and they're shifting that to the hip over here earlier in the stance phase. But we can go one level deeper down to the muscle levels. And now we can look at individual muscles and see what the differences in metabolic cost are across these biofeedback conditions. And once again, we see these muscles that are at the ankle joint. This is showing up in the joint level metabolic costs here for the medial gastrocnemius and the soleus. Lower costs at the ankle, higher costs at the hip during early stance. And so this confirms the joint work redistribution theory on an individual muscle level and solidifies that reducing propulsive force shifts the individual muscle metabolic costs from the ankle to the hip. So higher metabolic cost for walking when you use your hip more than your ankle and push off the ground with less force. Okay, and then I'll get into a little bit more of how I did this, just to kind of wrap this up. So I used a lot of batch processing and open source coding with OpenSim, and this is kind of like my pipeline from measuring the data into bringing it into OpenSim and performing these bioenergetic modeling simulations. And kind of making this into one big package really helped to manage all this data. Like it's quite a bit of data, especially once you start doing the simulations. And having it all in one place really helped to understand a lot of the modeling parameters. OpenSim has some really good tutorials where you can click through and kind of change this and that and see the effects within the simulation. But when you're doing it over and over and over again in a batch pipeline, you can see how just making one adjustment here really affects your results. And then lastly, it's a good way to build collaborations with others. I think after I started doing this, I started chatting with more and more students at my university and also kind of across the country. Be like, oh, hey, like, we found out that you're doing this and we're really interested in modeling. It was early in the pandemic and everybody was shut at home. And it's like, well, what are we going to do? We're going to model our data that we already have. So I ended up collaborating with a lot of folks, just giving them a like one-on-one on, oh, hey, here's OpenSim, here's what you can do. Be sure to, record this and use this data and that kind of stuff. And so the awesome thing about OpenSim is like they have APIs in both MATLAB and Python. And so you can kind of like pick your favorite coding pipeline and or coding platform and just code all day long and hopefully have something at the end of it. But there's also a ton of resources available on GitHub. And so I'm not the only one to try and do these batch processing scripts. There's probably a better pipeline, which is the batch OpenSim processing scripts, or BOPS, at that URL. And then also someone brought OpenSim into the Google Colab environment, which is kind of like a Linux-based platform that you can run Python on the cloud, and you don't have to install anything on your computer. So that seems like it could be pretty useful, especially for running a lot of simulations. And then some folks at Georgia Tech made these mocap tools that Lindsay told me about. Shout out to Georgia Tech folks, which are really helpful for like inputs and outputs, and also like setting up your own OpenSim processing scripts. So no one's starting off from scratch, and there's lots of resources out there for you to get started. Okay, and I guess I'll wrap it up by talking about some tips to go from experiment to publication. I think a big part is like going one day at a time. You can't do it all in one day. Like, don't leave it all for the last moment, and set goals and due dates while you're going through the process. One thing that I like to do is I like to code all my figures. So I make all my figures in MATLAB or Python. So all the data figures or results figures that you saw earlier in this presentation, those were all made in coding. And that helps if you want to add a participant or 7 to your data set, and you don't have to like redo everything in Adobe Illustrator or whatever figure generation software you use. It's a little bit more work up front, but it feels really nice to just like change one thing way at the beginning and just click run, and then all of a sudden you're pretty much good to go all over again with new data. You all saw some methods figures, and those help a lot when you're writing your methods. You can just kind of get a really good idea of, a solid idea of what you're doing and have it all in one image. And it really helps for writing. One thing that I struggle with is trying to make the first draft as good as possible before sending it off. But my advisor is always just like, just send me what you got. And it's, it's going to bleed either way, whether you spend 4 or 14 hours on it. So just, write it, have it done, and get more eyes on it. And then also like definitely take a few days off to reset, put it away for the weekends. Don't come back to it till Monday. Take a whole week off, having those days off to clear your head and come back with fresh eyes is really helpful. And that's all I got for this. So huge thanks to ASB and the student group for asking me to join for this. And I'm excited to hear your questions and have a discussion.
Good job. That was really cool. Sweet. Well, so then, as he said, the floor is open for questions. I know I got one or two, but if anybody else has some, definitely feel free to jump in, or if you'd rather me ask them, you can put them in the chat and I can ask them.
Yeah, so
go ahead.
Go ahead.
Nope,
okay.
You go.
Okay. So you mentioned you were given a set of data and asked to put something together in relation to biomechanics. This reminded me of what I'll be doing this summer. I'm going to be in a research position for the first time, and I'm slightly nervous about it because it's something that I've never done before. And so you mentioned a few suggestions and pieces of advice, but I was just wondering if there's anything else you may suggest to a first-time researcher.
Yeah, that's a really good question. I'm assuming that this is going to be at a new institution.
Yeah, so I'll be doing this through Boys Town National Research Hospital in Omaha, Nebraska. It's a neuroscience brain research program.
And one other question before I try and provide any advice was, do you have like a defined research topic and question?
No, so that's kind of the surprise about it. So I know about the program and I know the different things that I'll have to be doing. I'm under a mentor and they'll be giving me a set of data and I'll have to do something with it. And then at the end of the summer, I have to do a presentation. And just to give a little bit of context also, I'm a biomedical engineering student on the biomechanics and biomaterials track. So it makes sense, ASB. But yeah, so this is neuroscience and I'm interested in orthotics and prosthetics. So that's kind of the route I was going.
Okay. Without knowing too much about where you're going and what the question is going to be or research area is going to be, I would say like really spend a lot of time like getting to know your mentor and whoever collected or put together that data set, so that you don't have as many questions about what is this measure, how did you get it. If you have access to like a data dictionary for whatever data you're getting, those are super helpful because a lot of times you're just given a huge spreadsheet and you kind of have to infer like what each column is. So I would, yeah, just really make sure you understand what each data is coming in and like how they're all connected. If you have like a bunch of different streams of data coming in, like for biomechanics, like your EMG and your forces and your marker trajectories and your ultrasound, like maybe even drawing like a map of like, where is all this coming in? How can I wrap my head around all this new data that I'm supposed to learn and, do a project on in 10 to 12 weeks? It's a lot to ask, but definitely like having that big picture mindset in the first couple weeks could be really helpful.
Thank you.
Yeah, good luck with your project.
Thank you, I appreciate it.
To ask one that kind of piggybacks off of the same thing, I was just curious if the roles were flipped and you were about to hand somebody a data set that you had just collected, say there's a new PhD student or something, what do you think that you would do to make it as easy as possible? You kind of answered this a little bit, but what would you do to make it as easy as possible for them to hit the ground running, having been on the other side of that?
Yeah, that's another good thing to be thinking about, especially as people graduate and people move on to other jobs or take off for the summer and the project is expected to move forward. Having some really solid documentation of what you've been working on, what the goals are, where's your progress to date, is really helpful. And that's more of like a big picture thing, but having like the small picture for like that exact data set, where are the demographics files? How can you get those? Commenting your code is very, very suggested. Even if it's something as simple as like find speed, and then 50 lines later, it's find force. Even that can be really helpful. I also think like taking pictures and just dropping those into a document with a couple of sentences of like this is how you set up this piece of equipment, can be helpful, as well as if anything is password protected, making sure that you have those passwords saved elsewhere. We had this a lot at the Children's Hospital of Colorado. We had summer interns every year, and then also we just had a heavy turnover of research assistants and research coordinators because they were always getting into med school and PhD school and PT school and all that. So we got really good at, documenting our research progress and everything. But yeah, lots of Word docs, and it's kind of a pain to make it because it doesn't help the person who's leaving that knows all the stuff, but it really helps keep that project moving forward.
I find it helps too if you have to switch between projects, or like if multiple collections are going on and they're just like a little bit different, right? Like it's all motion capture for me, but like sometimes they're a little bit different. I was gonna ask on that one a little bit. Does anybody use MATLAB structures? Our lab went to this a while ago, and so I find that really helps. I like that much more than being like, okay, where are the 5 Excel documents I now need to make line up? Like, because you can stick the demographics in the same one, and it's like, here, this one's all about person 2.
Yeah, I love MATLAB structures. They're probably a big reason on how I actually got things to work and get this to get this study even done. I used a lot of MATLAB structures. 2 things about using those, as useful and helpful as they are. You can't really make any much bigger than 2 gigabytes. MATLAB starts to freak out in my experience. So if you're trying to save like all the marker data and all the force data in there, maybe don't, or like downsample it before you do that, because I try and save like 20 subjects worth of data to one MATLAB file and bad things happened. So don't do that. Or if you figure out how to do that, let me know.
Have you tried the dash, like the version change? There's like a version command, and I know part of it is it lets you make bigger files, but also to get around that, I just make each person their thing.
That's the workaround, is just make one for each subject. The other thing is, if you use Python for anything, it can be tricky reading in MATLAB data files into Python. Like, it doesn't make sense for a lot of it. Like, you can do it, but you end up having to do like the variable name and then like empty bracket 0, empty bracket 0, and then the next like subheading, and it's just a little confusing. But once you get it going, it works out pretty well. So yeah, love the MATLAB structures.
Cool.
My question's pretty similar. How do you structure your data, or like, do you have a way you keep data in different folders? Like, obviously you use structures once you get to MATLAB, but I've heard that how you structure your data before it gets to MATLAB also can make things more efficient, but I don't know more than that. Do you have a good format for that?
Yeah, how I've been doing stuff is you have like your study folder and then all of your subject folders within that. So folders times 20. And then inside of that, I have all of my biomechanics data, my forces, and my marker files in that. And then within that folder, I make OpenSim folder, and then I start saving all of the OpenSim outputs in there. And so it's like a really big hierarchical network that I'm sure someone has code to like analyze the file structures and draw a big tree, but I haven't found it yet. That tends to work really well. However, it can be difficult in MATLAB to like go through each one and like have all the naming structures right. In Python, I found a function called os.walk, which will literally walk through like every single file within a folder, and it'll like go to inside of all of the nested structures. So you can just say, like, make a for loop, like go through oswalk and like find anything that has activation _ force.sto and like save that path name, and then like start to do things to like bring that out of the big nested folder set. And that's been a huge lifesaver in the last year, but that's in Python and not MATLAB. So
Yeah, I get a lot, like my study's 4 days, and then I also have young subjects and old subjects, and so I'm really struggling with how to when do I actually need to keep things separate and when is it okay to have everything together? But I think some of it's just going to take some trial and error and figuring out.
Yeah, I know I have like two conditions like that too. And I like, instead of patient 1 through 20, it's like, it would be like young 1 through 10, old 1 through 10. And that would all still be on the same level of those, because like you're going to do the same thing to every patient or every subject. So that's, I split up that way. I have not been clever enough to find a MATLAB one that walks through all the folders. I just do nested loops and know which each level is.
Yeah.
I
The
agree
nested
there
loops,
should be a better way than that, but
yeah, the nested loops works pretty well, but once you start to get like 5 or 6 deep, it can be a little hairy.
I think I have one that like, it does it, but it's like just the loops and all it's doing is going in and pulling all the data out, and then boom, you have a structure, which is still like has that system, but like at least it's not in folders anymore. So
Yeah.
but no, that's cool. I had a different kind of question. I thought it was really interesting. Oh, unless Jenny's got one. Are you?
You can go ahead. I was just
Okay.
waiting for you to be done. Go
Sorry.
for it.
I was
Yeah,
gonna,
you're good.
I thought it was cool that you were saying that yours like reflects that what they were seeing before with the like moment shift. You're seeing it in the muscles, and that also then follows that you would increase the cost of walking. Do you have a hypothesis why that might be happening? I was sort of thinking about one, but I would be interested to hear.
For why the
For-
hip
for-
strategy.
Yeah, for
Costs
why
more?
the hip strategy costs more, or why, because like then they'd be taking shorter steps, so they're not going as far. So per stride.
Yeah, it's not my reason, but what I've heard and been told and what makes sense to me is that it has to do with muscle cross-sectional area and tendon lengths and stuff. So elasticity for the tendon, you have longer tendons at your calf because of the Achilles tendon, and so you can utilize the elastic storage and return a little bit more. Whereas at the hip, you have such short tendons that you can't really take advantage of that. And then also because the hips are so pennate, or, you know, all the muscles at the hip can be quite pennate, and there's just a lot more of them than what are at the ankle. You just have a greater volume of muscle being activated in order to do that.
Yeah, I was wondering, even that makes sense for sure. I was wondering even if it's just you have more mass, right? You increase the moment at the hip and you have to move the mass, more of the mass, right? If you're doing it all at the ankle, your foot is very small, but your whole leg is a lot to try and move through more.
Yeah, that's another one.
I
For
think
sure.
Art Quo, I think, might have a paper on that producing work distally is more energetically producing work proximally is more energetically costly than distally for the reason that you just said, Evan.
That might have been one of the ones I was reading last week, which is why it's in my head.
It's probably all of those reasons.
Yeah. You know, the mechanics people and the physio people sit in different camps but are saying the same thing.
Yeah, I mean that's good. That doesn't happen too often.
True.
I'll jump in now. Hi, Ricky. Good to see ya.
Hey Jenny, good to see you too.
Yeah, just had a question. So it looks like you've taken the dive into quite a few different batch OpenSim processing methods. And I guess I'm always looking for new ways because I had to get a new computer and now my OpenSim keeps crashing, even though it's the same script I used to run before, and my MATLAB's the same and my OpenSim's the same version. I have no idea. So I've like tried weird hacky things and changing out files until it works. But just wondering if you've had like any of the things you've tried have had a consistent experience with any, or anything that you would say is a little bit more reliable, because I'm down to switch to Python API if needed, because MATLAB's being a little bit temperamental.
Yeah, consistency can be difficult. So the thing about the Colab scripts and the Python one It looks really good on the surface, and I'm sure it works great. One thing just to be aware of is I think Colab cuts you off after like 10 or 12 hours. So if you have like a bunch of simulations running, they'll just like cut it right then. And then also like if your IDE interactive developer environment is not being used for more than like an hour or something, they'll also just turn it off. So you can't just like start it and like walk away and then come back and be like, okay, all my data's done.
Okay.
Which, I mean, that's kind of the reason why you'd want to use it, just to like run it and say, okay, 10 hours later, all my data is processed. The stuff on the computer is hard because if you have simulations running, you can't really do anything else with that computer, at least very efficiently. And it'll just slow down the simulations if you try and respond to emails or write your paper or do anything else. I've been using the computing cluster at UNC, which has been pretty nice, but it's also like a big activation energy requirement to like learn how to use that and get all the credentials and get your data up on there. And then some days the computing cluster is just down for no reason and you can't get on. I've had that issue. So like they all have like their own pros and cons, which is not the answer you want to hear.
So that's okay, as long as I can be a little bit more sane knowing that there is not a much easier way out there. But yeah, doesn't surprise me too much. I know our computing cluster has the same issue.
I had one that seemed like a reliability issue with the MATLAB OpenSim API. And then we finally figured out it was in MATLAB, you can't use any of the commands that are like just append. Like if you're trying to do something and append on the tool, it's just .append. That will gunk up the MATLAB, like, I forget exactly where it is, but so like if you went back through and like cleared your MATLAB cache by like going into the program and deleting some of the install files, then it would like work again for a couple and then crash. And so the trick we finally found was if you change all of your appends or all of your adopt and appends to clone and append, it has been going smoothly since then. So if there's a like control find option, I would see if clone and append helps. My only hypothesis for why that works is it's not actually changing an object, it's just sticking a new one in there, and MATLAB likes that better. But I did not major in CS, so I don't know for sure.
I will add a caveat for my stuff is that I don't even use the official API in MATLAB. I just set stuff up and then do system calls within MATLAB or Python and like change the XML file using some XML file changer function. And then just say, okay, system run this OpenSim-CMD-run tool-scale or whatever the tool is that I'm running. Because I never got the actual API to work right. And I was just like, we'll just override it. It's fine. And it works really well for Python stuff and MATLAB stuff. It's just different. So there's a lot of different ways to do it.
I'd say Ricky didn't mention it, but maybe this is how he was talking to all the people. The forum is pretty good for OpenSim. And it's not like if you ask a stupid question, like it's probably not stupid for A, but like B, they do reply, and like their response, if they deem it a stupid question, is to just reply with the link to the topic on the forum that has your answer in it. So like, I don't think there's like, nobody's like, if you do it and it's a stupid question, then everybody knows who you are and that you asked. Like that does not happen at all. So like, especially if anybody has to move to Moco, that one's real responsive. And there are even some other PIs around the country that are very religious about checking it and answering questions. So it's open source, so it's hinky, but like they do have a forum they still respond to.
Yeah, the community is really great. And I've had good luck with the forum as well. And also, like, it's not a stupid question because someone will probably look for that question like pretty soon after you and be like, oh, sweet, this person just asked that, and here's the link, and now I can go there and figure it out.
Or if you know the answer, like write it up, or when you figure it out, add it to the end of your thread so somebody else can look at it. Definitely replied on the ends of one, like, so did you figure it out?
I've honestly thought about with some of the recent OpenSim machines I've been having, posting and immediately responding with the like resolution that I figured out, just so it's like on the internet somewhere. Because yeah, some niche issues like this pop up with the API that I just can't ever seem to find on the forum.
I think there was one that I figured out and answered so that I could go back and search on the forum and find it there. Even if no one else does, I can go find it there. Oh, cool. Go ahead, Kristen.
No, I was gonna change the subject if that's okay, and ask kind of a meta question on the biofeedback. I think at least in all the research that we do in our lab, we're usually giving some sort of biofeedback to the participant. How much do you think the type of biofeedback and how well we give it is actually influencing our results and our studies?
I will answer the how we give it first, and then we can get into the type. Not for the study I talked about today, but the last study I did, I made a game out of it, and we tried to we gave them like a streak counter when they got like on the target for multiple steps in a row, and like to see how many steps in a row they could do it. And I think with some participants, like towards the end, they're like, no, like I don't want to stop. Like I'm almost like I was hitting a new PR, or like I was on streak and like this was going awesome. Or maybe I was just thinking that. So, I mean, it's obviously going to influence the results because that's what it's supposed to do. It's biofeedback, and you're asking them to change some things so you can measure the change in results. So there's that part. The type, yeah, like the visual is the easiest one to do because we can just code that in and make a figure and show them where they're at. I've seen some cool studies on hearing, and also some, maybe not studies, but just some groundwork on like haptic stuff for like vibrations when you hit a certain threshold. I've seen other forms of biofeedback where like with upper arm stuff, and you're like manipulating a robotic hand, but you can't see what you're grabbing. And like they give you like haptic biofeedback with vibration, like on your brachial nerve over here. And you're supposed to try and guess if you're picking up or if you're holding a sphere or a cube. And for a lot of that, it's just like you can't really like figure out why you think it's one or the other. You just know. It's kind of like machine learning. Like, okay, like I felt like this when I picked up a sphere in the training, but now I have no idea. And I guess it kind of feels more like A than B, so I'm just going to go with A, but I'm not exactly sure why. So the haptic stuff can be really, really strange. The auditory stuff could be really interesting as well. And I just like started to think about like when your headphones short out or like something really loud comes on, you just like jump. Like what's causing that? Like is that pain receptors? Like what's the neural pathway there that makes all your muscles contract? And so yeah, maybe we just came up with a new study on auditory feedback and scare tactics, I guess. Bye, Claudia. Nice to meet you. Good luck with your summer internship.
Thank you.
Yeah, I guess I just think about it too, like you said, is like some of your participants were obviously taking the feedback and using it as a game and wanting to beat themselves and getting competitive about it, versus if you didn't gamify it in the way that you did, would they have performed the task as well as they did, which is obviously then going to influence your results. Mm-hmm. And how much of that, like we go read a study and you're like, wow, they found a 15% difference and they were using this feedback, if they used a different feedback, would they have found a 30% difference or they found a 0% difference? Like, I feel like we're not in a space yet where we can answer that question. It's also really interesting because we, at least in our lab, I would say the feedback aspect is not the biggest priority in terms of the experimental design side of it.
Yeah, I guess in terms of the design side, like you just want it to be functional, but like we're not working on immersive virtual reality environments.
Right.
Like we're not getting into the metaverse or whatever we call it these days yet. It's interesting to think about, but like we don't have the time to go into that and get to that level. But also, like I guess the question I have coming back to that is, are we measuring biomechanics or are we measuring behavior?
You're doing both, especially if you're giving feedback.
Yeah, but like which are we like grounding our results in biomechanics or human behavior?
I don't think that you can fully separate them, especially if you're thinking about like learning studies or even on a push off feedback study. They need to do the behavior the way that you think that they're doing it in order for your biomechanics to make sense. Yes, we're analyzing all the biomechanics, but at the same time, we need their behavior to match what we want it to be, or we need to at least be able to explain their behavior and account for it, even if they're not doing exactly what we want them to do.
Yeah, I completely agree with you. I think biomechanics is behavior and behavior can be biomechanics. And yeah, one version of biofeedback might be better or worse than another in terms of creating a response. And maybe we need to do more work on exactly how that is. And there's just so many other variables in that in terms of behavior. Like, you know, what are they primed to do? Like, is you know, what else is in the lab? Are there things that they're focusing on? Are there things that make them more or less interested in this walking study and a line on a chart in front of them for 5 minutes?
Right. Like, are they competitive? Are they not competitive? Are they inclined to do well on the task? Do they care? Like, I can't tell you how many old adults were like, oh my God, I'm ruining your study. I can't do it. I can't. They're like, no, you're fine. Like, you're trying to do the task. That's really all I ask. But like, and then other ones were just like, oh, I don't really care. I'm just going to do the study, like stuff like that. So like, there's that whole aspect of kind of the psychological side of our studies that you never hear talked about in our papers, but I find it super fascinating.